Emily Shields and welcome to the Cross Report. A monohybrid cross is a mating between two different alleles at a genetic locus of interest. Monohybrid crosses were discovered using garden peas and corn between 1882 and 1884 by Gregory Mendel, observing the phenotype of following generations. His experiment resulted in the discovery of the law of segregation, paving the way for genetic modification today. To explain further, we now go to Harry in the Powtoon room.
Mendel, yes, that guy, was an Austrian monk and a pivotal figure in monohybrid crosses and our understanding of evolution. Mendel disproved previously held theory created by Aristotle that offspring are a mixture of their parents' characteristics, replacing it with his law of segregation derived from his experiments published in 1886, Experiments in Plant Hybridization. Mendel, yes, that guy in the veggie patch, experiments took a pea plant seedling allele, two forms of a gene arising from the same mutation, e.g. plant height, by breeding two opposing of the same characteristic, tall times short, and observing the genotype expressed, which is known as the phenotype. To express this process simply, we're going to use Punnett squares invented by English geneticist Reginald Punnett.
Mendel took a homozygous dominant plant represented by big T, and bred it with a homozygous recessive plant represented by little t. Dominant meaning it is expressed over the other gene, in this case tall is dominant. As depicted by the Punnett square, the F1 generation will produce a tall phenotype with heterozygous genotype big T,
Mendel discovered the law of dominance, that one gene, in our case big T, if present in the genotype, will be expressed in the phenotype as a tall plant. He recognized the mathematical pattern of inheritance and allows biogenetics today to predict future generations of Mendelian traits and produce a desired phenotype in the interbreeding of specific alleles. An Australian example of a monohybrid cross is Drosophila melanogaster, or the common fruit fly. We are focusing on the allele that controls the thorax colour, a Mendelian trait in which a light thorax will be represented by big L and is dominant, and the dark thorax, which is recessive, will be represented by little l. If the lighter thorax is represented by big L, heterozygous, and the darker thorax is represented by little l, homozygous. As shown below in the Punnett square, the F1 generation will produce 50% light heterozygous phenotype and 50% homozygous dark phenotype. A utilisation of monohybrid crosses to benefit broader society is transgenic wheat plants that are herbicide resistant. The resistance relies on the expression of a recessive bar gene represented by little b. The non-herbicide resistant gene is represented by capital B. Therefore, by breeding two heterozygous big B little b wheat plants, herbicide-resistant wheat will result. 25% of the F1 generation is herbicide-resistant, as depicted by the Punnett square. As more generations are bred, the percentage of little b homozygous is increased. This allows for faster and more cost-effective protection of wheat crops against weeds. Mendel, dubbed the father of genetics, built the foundation of our knowledge of hereditary biology from his finding in experiments in the 19th century. His discoveries and laws have been used to solve monohybrid crosses and produce GMO crops to benefit broader society. Thank you, Mr. Mendel. Thanks, Emily. That's my explanation of Mendel's monohybrid crosses. Back to you at the desk.
Monohybrid crosses were a pivotal biological discovery by Gregory Mendel and have been used to solve genetic problems to benefit society. I'm Emily Shields. This is the Cross Report and...